Good afternoon, students. In this video tutorial, we'll be going to see about transduction in bacteria. So let me start this session with the question, right? What is transduction actually? What transduction? Right. This is the question. And in the previous two lectures, we have discussed about the two important phenomenon in bacteria called conjugation and transformation. In both the cases, you might have studied that how genetic traits, particularly how new genetic traits are being spread among the bacterial population through this mechanism like conjugation and transformation. This is another important phenomenon in which bacteria acquire new properties, new genetic traits, which may be um, galactosidase enzyme production or capsule formation or antibiotic resistance. So, these are all few examples, and all these are called as genetic traits, genetic characters, and these characters are acquired by the bacterial cell through the virus, right. So, if genetic traits are transported between bacterial population with the help of virus, then the mechanism or the phenomenon is called as transduction, right. Remember, so in simple words, transduction refers to the transfer of genetic traits between bacterial population by a virus, typically a phage. In this case, bacteriophages are key role players, right. So, with this basic idea, we'll move on to the next slide. And you probably studied the architecture of a virus, I mean specifically about bacteriophages. So, viruses which feed on bacteria is said to be bacteriophage, right. Right. So, what is the name of a virus which feed on fungi? Exactly, the answer is mycophages. So, what would be the name for a virus if it feed on animal cells? Oh, very good, very good. Nice to hear the answer, and the answer is zoophages, right. So, here we are going to see about viruses that feed on bacteria. So, they are called as bacteriophage, right. So, one such example is given here. A typical bacteriophage has a complex head, right, which encloses the genetic material. It is called as capsid, a protective layer which encloses the genetic material of the virus. So, I have explained here. Remember, this is the structure of phage head which containing the nucleic acid. So, at the base of the head, you could see hollow stylus, a tube that extends from the collar of the bacteriophage. So, at the base of the stylus, you could find the base plate, right, the what's that purple color layer. So, below the purple color layer, you could find pins called tail pins, and tail fibers are the basic structures that makes up mature virus particle or virions, VIRIONS, called virions, right. Remember these terminologies or these glossaries which will help you to write the 2 mark questions, right. So, what are virions? Virions are mature virus particle. So, what is mature virus particle? A virus particle with all structural components, right. And this is the architecture of a bacterial cell. You probably know about it. So, I do not want to waste much of time in this slide, right? Right. So remember, you are growing a bacterium in a laboratory within a nutrient medium, right? And here the bacteria is surrounded by numerous virus particles. Numerous virus particles. How it is possible for a virus to grow in a synthetic media? Because we know that viruses are obligatory intracellular parasites. It can grow only in the presence of living cell. Remember, here the bacteria is a living cell, so it is called as bacteriophage. Bacteriophage. So these viruses are not living in this environment by utilizing the chemicals present in the nutrient medium. Rather, it depends on the bacteria, right, and multiplies, right. This is the principle, right. No virus can grow in a synthetic medium. It definitely require a living cell for its survival. Remember this point. And how a virus identify its host, because there are different types of bacteria growing in a microenvironment, right. There are several different types, and how a virus particularly identify its host. We already know that virus exhibit a phenomenon called what? The answer is trophism, right. Trophism is specificity. Remember, trophism, the terminology trophism is specificity. And how a virus, typically a bacteriophage, identify specifically its host, for example bacteria, right. So by random collision, right, by random collision, simply by random collision, no other means, a virus can identify its host. See, a bacterial cell has plenty of receptors on their surface. One such receptor is suitable for a virus to absorb, right. Remember, these inverted triangles are mentioned here as the receptors. So upon random collision, a virus can bind with the receptor so that it identify its host correctly, right. Not all the viruses are absorbing on the surface of bacterial cell. So after binding with the receptor, right, see, now the binding is completely established by utilizing the receptor expressed by the bacterium on the surface. So after adsorption, right, and here you could see uh, a small thread like structure. Let me zoom it, right, a small thread like structure. And what does it mean? Remember this area, right, this particular point. I want to conclude, I mean, I want to focus it, this particular point, a small tube like structure that pierce the cell wall and reach the cytoplasmic membrane. Once this layer senses the presence of a chemical called phosphatidylcholine, right? Phosphatidylcholine. Once it recognizes, I mean, once the phage tail recognize, tail in the sense the hollow stylus recognize phosphatidylcholine chemical, right? Once it is sensitized by the phage hollow stylus, the head of the virus started to constrict and release the nucleic acid into the bacterium. Not a simple mechanism it is. It is a very complex mechanism, right? And the signal here is phosphatidylcholine, a chief chemical component present in the cytoplasmic membrane of bacteria. And this chemical will be recognized or sensed by the hollow stylus of the phage. Upon recognizing, right, upon recognizing this chemical, the phage head constrict, thereby injecting the nucleic acid into the cytoplasm of the bacteria. Right. Now we know that the phage DNA is inserted into the cytoplasm. Right. So upon absorption, the injection of nucleic acid takes place. And in the next slide, a typical phage genome has two essential components, right? One is early gene, another one is late gene. These two genes are the key role players, right, in the multiplication of phage. So, what is the role of early gene? Early gene is known for synthesizing enzymes required to destroy the host genome, right? It is essential to destroy. It is essential to destroy the host genome, right? I'll draw it again. It is early genes are known to code for enzymes that destroy the host genome. And what is the role of late genes, right? So, late genes are known to synthesize phage specific structural components. So, what are phage specific structural components? For this, let's go on to the previous slides, right? Here you could find the structural components like head, uh, like collar, like hollow stylus, base plate, tail pin, tail fibers. All these things are structural components, and these proteins are synthesized by the late genes of virus nucleic acid, right? So, let's move on to the next slide. Once the viral nucleic acid is inserted into the cell, right, I mean particularly the bacterial cell, you know that viruses are molecular hijackers, particularly the bacteriophages are molecular hijackers. Why they are hijackers? So, upon entering into the bacterial cell, right, I will write here molecular hijacker, right. So, upon the entry, a virus started to utilize soon after the entry of nucleic acid, it started to utilize the host machinery to synthesize its own proteins. Machinery, what I am meaning here is the enzyme machinery, right? It started to use the host enzyme machinery to synthesize virus specific structural proteins as well as essential proteins required for its multiplication. So, here it uses host RNA polymerase enzyme to transcribe the genetic material so that mRNA will be synthesized. This mRNA is transcribed from viral nucleic acid. And in the next slide, and upon utilizing the host translation machinery, I mean the ribosomes, right, the viral nucleic acid is translated, right, remember, because viruses do not have any membrane bound organelles such as ribosome. Bacteria has its ribosome, but in bacteria it is called as polysomes, right, and these polysomes are being exploited by a virus for the synthesis of its own specific structural components, right, remember, because viruses don't have any such machinery. It don't have enzyme machinery, it don't have ribosomes, right. All these things are utilized by or utilized from the bacterial cell. So, that is why they are called as molecular hijackers. And remember, and here I have given two compartments. So, what compartments? So, remember, you can split this. Remember, this is a bacterial cell, On one side, on the left hand side, you could find virus components. So you could find head, right? You could find hollow stylus, you could find collar, right? You could find tail fibers, everything. All these are structural proteins of virus, right? Virus agents. So on the right hand side, you could find uh, disintegrated host genome, right? So this is a simultaneous action. Early genes are and late genes are parallelly expressed. So that early genes, we know that it destroy or it completely denature or it completely uh, slice all the host genome into small pieces. So this is what I mentioned on the right hand side of the presentation, right? This is uh, disintegration of host genome. On the left side, I have explained the synthesis of structural components of virus, right? And all these structural components are started to assemble. The process is called assembly, and all these things are assembled within the bacterial cytoplasmic region. So the individual components like head, hollow stylus, collar, tail fibers, so all these things are assembled so that a mature virus particle will be formed, and this is said to be a virion. We already discussed this word virion, mature virus particle. This mature virus particle has all its structural components. So upon assembly, it started to here you have to notice one important thing is while packaging, while assembling its structural components along with viral nucleic acid, sometimes probably a part of bacterial genome may get integrated into the virus head, right? This is actually called a generalized transcription, generalized transduction, right? Generalized transduction. So what happens during generalized transduction process? Upon the lytic cycle is going on, phage particles are synthesized and started to assemble in the cytoplasmic region, and during the assembly process, sometimes bacteria taken up, sorry, the virus particle taken up a small fragment of bacterial genome. So one such phage you can see here, the head has, right, a copy of this encircled phage has a copy of disintegrated viral, bacterial nucleic acid, right? The encircled phage, just look into the head, it has a fragment of, a disintegrated fragment derived from bacterial genome. This sometimes happen, right? This is actually a rare circumstance. It is called generalized transduction process, right? And after this, the virus, the population of virus inside the bacterial cell is keep on growing in the sense keep on increasing in numbers. At one fine time, the cell started to rupture. The bacterial cell started to rupture, right? This process is called assembly. And another important terminology associated with this stage is called burst size. Burst size refers to number of phage particle yield per bacterium, right? This is called burst size. And upon multiplication of the phage and assembly of the phage inside the bacterial cell, the cell beyond the limit it started to lyse, and through the lysed structures the virus will come out as a mature infectious virus particle. So you could find here, right? So through these areas and through all these gaps, right, these mature virus particle will come into the area neighboring cells. And upon finding upon release, right? So in this slide we discussed about how a virus particle is coming out of the bacterial cell. So upon exiting the dead bacteria, right, it is ready to infect the neighboring cell, neighboring bacterium. But here you have to remember one important thing. It is this phage, not all the phage, those phages which accidentally take up a fragment of nucleic acid, right, in their head started to infect the adjacent cell, and this is called a defective phage. This is called a defective phage. And this phage now is able to transfer the genetic material from one bacterial population to another bacterial population, right? So this is what I told you. This is also a type of transfer of genetic traits between bacteria, right? Bacteria 1 and bacteria 2, the population may be different, but virus is acting as a tool to transfer the genetic material from one bacterial population to another bacterial population. This particular DNA may be code for novel traits like heavy metal degradation, synthesis of industrially important enzymes, capsule formation, which is an added virulent nature to the bacterium, right? Antibiotic resistance, whatever the things may be, and whatever the genetic trait may be transferred by the virus particle to a adjacent bacterial population. And this entire process is called generalized transduction, because whatever the fragment, whatever the piece of fragment may be uh, accommodated by the virus head during the time of release and assembly process, and followed by mm, this procedure. I mean, there is another kind of transduction called specialized transduction. So for that you have to understand what is lysogenic cycle. So the lytic cycle, you know that at the end of lytic cycle, the bacterial cell will be completely or irreversibly destroyed, so that mature virus particle will comes out of the bacteria. You know that this is what happen in lytic cycle. But in lysogenic cycle is very different. So upon injecting the nucleic acid, right, virus nucleic acid into the bacterial cell, this nucleic acid undergoes recombination in specific areas of bacterial genome, and this is called a site specific recombination by integrase enzyme, right? So this, the process of integration of viral nucleic acid into the host genome is mediated by the enzyme called integrase, right? Integrase. Fine. And now the bacterial genome is having a copy of viral nucleic acid in the integrated form. This is called prophage. This is called prophage. Prophage, right? Prophage, right? And now there are chances for the excision of the integrated copy of viral genome. Excision in the sense the viral genome may get separated from the bacterial chromosome. So what are all the conditions that favors the process of excision? Here I have given three process. One is UV exposure, second one is hydrogen peroxide treatment, third one is nutrient depletion. The process of excision of viral nucleic acid from the bacterial genome is said to be lysogenic induction. Lysogenic induction, right? So upon the process, upon induction process, right? So this, if it remains, if the condition favors, the bacteria still carry a copy of viral nucleic acid for many generation, right? For fifth cycle of generation, or for 10th cycle of generation, or 100th cycle of generation, right? The condition goes on without any disturbance. If the condition is not favored, right? Remember, either the bacterial cell is exposed to UV light, or may be exposed to hydrogen peroxide treatment, or may be exposed to a condition where the nutrient level is very poor. Under these circumstances, the viral nucleic acid gets excised. The process is called a lysogenic induction, right? So upon excision, it suddenly it enters into lysogenic cycle. It enters into lysogenic cycle. Sorry, lytic cycle, right? So upon excision, it enters into lysogenic cycle, right? And here comes the major turning point. So what is the things to be noted? So we know that upon excision, uh, the lysogenic cycle is converted into lytic cycle. We know that, right? So this is what happened. But generally, the excision is very formal, very precise. But under some rare circumstances, during the time of excision, a copy of bacterial genome may come along with viral nucleic acid. In some other words, a portion of viral genome may still attached with bacterial chromosome. A part of the viral genome along with bacterial chromosome excised from the integrated form. Most of the time the excision is very precise, right? Just like GPS guided missile, right? It will fix the target and destroy the target correctly without destroying the nearby areas. In the same way, excision is very precise mechanism, right? Under some circumstances, under rare occasions, along with during the time of excision, right, the viral genome excised along with a fragment of bacterial chromosome, right? Fine, but this is very rare alone, right? And in the right, so this is called a hybrid genome, right? This condition is called a hybrid genome because this is viral nucleic acid, viral nucleic acid, and this is called, this black area is called bacterial chromosome, right? So I told you, if the excision is not precise, sometimes a fragment of bacterial DNA comes along with viral nucleic acid. So upon excision, it enters into the cycle switched on to lytic process, and upon the synthesis of virus particles, this hybrid genome may get integrated into the virions, into the mature virus particles. See, you could find the presence of hybrid genome. Why it is called as hybrid genome? We know that it has a portion segregated from bacterial genetic material. So it is called hybrid genome. Now this defective phage is capable of infecting the neighboring bacterium, as what we saw in the case of lytic cycle, and integrates this defective genome, the hybrid genome, into the bacterial cell, right? Insertion of this hybrid genome into the bacterial chromosome. And this, see, you could find here the presence of hybrid genome in the cytoplasm of bacteria, and this will undergo recombination into the host chromosome in specific locations. This is called site specific recombination, I told you earlier, and this recombination enable the organism to acquire new property. In other words, the host organism, particularly the bacteria, uh, undergo a permanent change in the genetic makeup, right? A permanent change will happen due to the integration of hybrid genome into the bacterial nucleic acid, right? And this process is actually called specialized transduction. And here, the in many textbooks, the example given for a specialized transduction is the transfer of beta galactosidase gene from one organism to another organism by a virus, right? The recipient organism still, for example, the organism is going through 5 generation. So, still fourth generation, the bacteria lacks the property to synthesize beta galactosidase. Upon fifth cycle, followed by virus infection, the bacteria acquire the new property or the novel property of synthesizing beta galactosidase enzyme. So, thereby the bacteria acquire new or novel traits due to the infection of virus, and this phenomenon is collectively called as transduction. Transduction. There are two types of transduction. One is generalized, another one is specialized transduction. Remember, stay tuned with me. Still, if you have any queries or doubts, don't hesitate to ask me, right? I am here to clarify your doubts. Thanks for listening. Bye bye.